All right, guys, we're going to start on time. This is pretty good. So I am Barb Slusher, and I am a professor at Johns Hopkins. I also run Johns Hopkins Drug Discovery, which is a team of folks that do drug discovery based on academic inventions. I'm also the vice director of Brain Science Institute at Hopkins and also co-direct a center that does development for neurotherapeutics, HIV specifically, neurotherapeutics. Before I came to Hopkins, I spent 18 years in the pharmaceutical industry doing drug discovery, and so have basically experience doing this both in a pharma as well as in an academic setting. And so what I'm going to talk to you today is a little bit different than the other lectures. So the other lectures were focused on the operation of drug discovery, teaching us about target validation and screening and chemistry and pharmacokinetics. Today, I'm going to talk to you, give you some examples of doing drug discovery in academia. So I have two topics that I want to cover. The first is the ecosystem of drug discovery over the past 2 decades has really changed, and academia is becoming more involved in doing drug discovery. And so I want to talk a little bit about that change in ecosystem and the rise that academia is playing. And then the latter part, I'm going to give you 3 vignettes, 3 examples of drug discovery. The first one is going to talk about how we can have innovative public-private partnerships in drug discovery. The second is taking an idea from the bench all the way to the bedside in academia. And the third is turning a really failed project into a successful project. And so I'm going to walk through each one of those. All right, so first, the ecosystem of drug discovery has really changed. And I'm just going to give some changes in pharma, the NIH, and then in academia. So I think in pharma, at least my experience in pharma during the 1990s and early 2000s was that pharma was in my experience during that time, was very closed, secretive, intramural. Everything we did, we didn't talk to people about. It stayed within the company. And I think that's changed dramatically over the past decade or so. I think in the 21st century, farmers have been much more openly innovative, collaborating with each other, or sharing risk with each other, as well as working with academia. The other big change is NIH. NIH has become increasingly interested in doing translational research. So under Elias Zerhouni, we started the NIH roadmap, which was focused on translational research, and then more recently, under Francis Collins, an entire institute focused on translational science. The NCATS, or the Center for Advancing Translational Science, was initiated, whose focus is to transform the nation's medical research capabilities into improving translation of those research into new therapies. And so again, this has been a difference or a change in NIH. All right, and then let's talk about academia. In academia, there's been a really dramatic increase in the number of academic medical centers that have drug discovery or translational centers. And so this is a picture of the United States. I'm sure you guys could have figured that out by yourself. But this is in 1990, and these little red dots here are showing you the academic medical centers that had translation or drug discovery. And so in 1990, there were 6 of them. In 2002, there was about 20, and if you look now, there's over 100 academic drug discovery centers or translational centers. So this, over a pretty short period of time, is a big change. Now, not only do we have more academics doing translation, but there has been a really dramatic increase in academia and industry working together to partner on drug discovery. And so I'm showing here from 2002 to 2022, so over 20 years, there's been a 16-fold increase in academia and pharma partnering together on translation. So again, these are pretty dramatic changes over a short period of time. Not only are we seeing academia working with pharma, but also the venture world. And so if you look at how many investments, money is being put into startups from academia, that also, it's a 2,000% or 20-fold increase over this 10-year period. So I just wanted to start with that as sort of the backdrop to say that this world is changing in drug discovery, and which is part of the reason why we have this course to teach academics about the process of doing drug discovery. Okay, so that was kind of background about the rising role that academia is playing, I'm going to spend the rest of the time giving you 3 vignettes about doing drug discovery in academia. Okay, vignette 1. Vignette 1 is going to focus on system XC. This is a transporter in the brain that we have been looking at for brain cancer. And hopefully this is going to be an example of how pharma and academia can partner together in drug discovery. This is a collaboration that my team has had with the pharmaceutical company Eisai. All right, so a little bit about this target. This is, system XC is a cysteine glutamate antiporter. What does that mean? Well, that means when it takes cysteine in at the same time pumps glutamate out, okay? And this is important because this is important in the cellular uptake of cysteine, which then is oxidized into cysteine, which is really important in synthesizing glutathione, so involved in redox control. At the same time, this is involved in non-vesicular release of glutamate outside the cell. And so this has been shown, this transporter, the system XC transporter, has been shown to be upregulated and involved in brain cancer, and specifically in the involvement of promotion of brain cancer. And so, let me see if I can. So here, I'm not very good at drawing, so this is as good as it gets. So this is a normal cell, and this little red here is showing is the system XC. And so what happens is this system XC, under normal conditions, brings in cysteine, makes glutathione, kicks out glutamate, then glutamate is taken back into the cell by these glutamate transporters. Okay, that's normal. Now what happens in glioma? So in glioma, what happens is the XCT doubles or triples. So you're seeing much more system XC. And so again, bringing in cysteine, dealing with redox potential, but then glutamate, you're getting much more glutamate being kicked out. At the same time as glutamate's getting kicked out, the glutamate transporters are being downregulated. Okay, and so what happens, this results in more glutamate being released, less glutamate taken back into the cell, which is leading to this high extracellular level of glutamate outside the tumor. Okay, and this causes seizures. It also kills the surrounding tissue so that the tumor can grow. So if you think about this from a tumor perspective, this is super smart, right? So you're bringing in cysteine, you're making more glutathione, you can deal with the redox, which is really important for rapidly growing tumors. At the same time, you're kicking out glutamate and killing everything so you can grow, right? So from a tumor perspective, this makes a lot of sense. And because of that, people have been interested in this as a therapeutic target for brain cancer. So these are several pretty high-profile papers that were published in Nature Medicine and Journal of Neuroscience, showing that in mice, that if you inhibit system XC, either with a pharmacological inhibitor or genetically, you can decrease this peritumoral glutamate, you can decrease seizures in the animal, you can decrease tumor volume, and you can also extend survival. So if you're a mouse, this is validated. If you inhibit this target, it is useful for brain cancer. In patients, there's also was a more recent Science Translational Medicine paper that showed that in patients, if you have high levels of system XC in your tumor, that is associated with higher peritumoral glutamate, more seizures, and a poor prognosis. So what's showing you here is that patients that have high system XC die earlier than patients that don't. And if you look, you can see that the tumors that have the high system XC also been able to show that they have high peritumoral glutamate, which is likely causing their increase in seizures. Okay, so this is a validated target in mouse and also clinical evidence. So we wanted to look at this from a drug discovery. Can we make a system XC inhibitor? And there's really nothing out there. The two pharmacological tools that people use are lousy. Okay, the first is sulfasalazine, it's 30 micromolar. S4-CPG is another, it's 15 micromolar, both weak. Sulfasalazine itself is extensively metabolized by azoreductase in your gut, and the two metabolites that are generated don't inhibit system XC. So in vivo, this is a tough drug to use. And S4-CPG is actually a better mGlu-R agonist than it is a inhibitor of system XC. So, and in addition, both of these drugs have carboxylates, which make it difficult to get to the brain. So these compounds are available, but they're not very good. And so our first effort was, could we Could we make better analogs, more brain-penetrable analog? Could we make them more potent? And I'm going to spare you all the details. No, we could not. We failed. And so we discontinued this effort. And so the next thing we thought was, well, let's start over again. Let's see if we can do a large, unbiased screen, high-throughput screen, and see if we could get some system XC inhibitors. And so that's what we did. So Ajit Thomas in the lab established this high-throughput screening assay. It was a fluorescent assay based with astrocytoma cells. Basically, very simple. You put cysteine in, it goes in, and glutamate comes out, and then we measure glutamate by a series of oxidation reduction reactions. The assay was performed very well at a good Z prime, and so we had this assay. So we're like, okay, now, what are we going to assay? So in pharma, you have these big, large chemical libraries that we can assay, but we're in academia. We don't have the access to the same types of libraries. And so I started talking to companies and saying, will you work with us? We have this assay. Would you be interested in collaborating with us to do a high-throughput screen? And so a lot of people said no. I didn't put them on the slides because that's not exciting, but we did get a taker. And this was the company Eisai. And there's a lot of words on the slide, but basically, we executed a high-throughput screening agreement with them whereby we bring targets of interest. Okay? If they're interested in those targets, then my group develops a high-throughput screening assay. And then we transfer that to the company. And the company screens through their million compound library. Any hits they get comes back to my group. We do the drug discovery effort, the SAR on those, and then the Eisai, in this case, has the ability to license it back in. All right, to me, this is a total win-win, right? From an academic, this gives us access to a good drug library. From a pharma perspective, this gives them access to a target that they wouldn't have otherwise gone after. So I think this is a real win-win. In fact, when we announced this, it was a deal of the week in the pink sheets. Okay, so how does this partnership work? I always get a lot of questions, so I put this slide in last night. So this is how it works. We disclose the targets. So everything in yellow here is academia, everything in blue is company. So here we disclose the targets, the company says which targets they like, and then we develop an assay and then transfer the assay to them. They run that assay through their large chemical library. The hits that come out, we get this hit list, and we can either in collaboration with them or on our own do structure-activity relationship, basically drug discovery to come up with a hit. If we get it to a lead series, we can license it to them for $1. If we take it to candidate selection, we can license it to them for $5, so it's all proportional. I made the $1 and $5 up. You guys probably realize that. But this is basically how it works. There's a list of criteria, and if we get to a certain stage, they can opt in. Further stage, they can opt in. And so we did this, and 2 years later, we had screened. We got some leads from that, did SAR, and we got a hit. The lead series. And this compound, unlike the two I talked about before, is actually nanomolar potency, not micromolar. Its potency in this series correlated with the inhibition of glutamate release, and it was well behaved. It was quite orally bioavailable in 2 species and brain penetrable. We had a plasma to brain ratio about unity. So this is looking like a drug. And so what happened, we identified this and we licensed it to the company. And so this has gone back to Eisai, and they are now moving this forward in development. And so anyway, that's vignette 1, and hopefully I've convinced you that there's some positivity for pharma and academia to be working together for the benefit of this drug discovery project, ultimately, hopefully, for patients. Okay, let me move on to vignette number 2. So vignette number 2 is focused on tumor-targeted antagonist of glutamine. And this is an example of going all the way from an idea in academia to getting it into clinical trials. And this is a collaboration, again, with my group and the cancer immunotherapy team at Hopkins. And so let me tell you about this story. So this story is based on a drug called DON, which is diazoxynoreleucine. And DON is a broadly competitive glutamine antagonist. And so here's a structure of glutamine. Here's a structure of DON. They're exactly similar, except glutamine has this amine group and DON has this diazo. warhead. And so this is a naturally occurring compound. It was identified from bacteria in Peruvian soil back in the 1950s. And it was shown at that time to broadly inhibit all the enzymes that utilize glutamine, right? Because it looks like glutamine. So it binds to those enzymes and inhibits it. And so we all know glutamine is absolutely critical nutrient for tumors to grow. And so people had shown that this compound DON could block tumor growth. And so and I'm showing in a diagram from a recent review we did, this glutamine, it's not a single enzyme. There's multiple pathways that this DON inhibits that are glutamine-requiring enzymes involved in nucleotide synthesis, coenzyme, glutaminolysis, amino acid synthesis. So it's selective for glutamine, but inhibits multiple glutamine-dependent enzymes. And so the data preclinically was so robust, this got taken into clinical trials back in the 1950s and 60s. There's probably about 15 clinical trials that were performed. And it worked. It showed anti-tumor activity. The problem was, go the other direction. The problem was that it wasn't very tolerated. All right, and the dose-limiting toxicity was gut. It was GI toxicity, nausea, vomiting, and gastritis. That's because your gut is highly dependent on glutamine. And so we got involved in this project with the thought that, what if we could take this active but toxic drug and make a prodrug of it, which would deliver it to the tumor and not to the gut, so that we could expand that therapeutic index. So that was the idea that we started about 5 or 6 years ago. Does that make sense? Make a prodrug, itself is inert, make that prodrug get released in tumor, not in gut. And so this is a schematic of our idea. So here's the structure of DON. It's got 2 easy handles on it, this carboxylate and this amine, where we put the promoieties. And so the idea was we would put promoieties here that would be preferentially clipped off by enzymes that are enriched in the tumor, but are not enriched in your gut. Okay, and so the idea would be we'd give this prodrug, this DON active with the promoieties on it. It'd be inert, inactive, it'd circulate that way. But when it got in the presence of the tumor, which has these tumor-enriched enzymes that would take the promoieties off, it would be activated. Okay, so that was the basic idea that we had. And so an ideal prodrug would be one that was stable in your plasma, stable in your liver, certainly stable in the gut, right? We don't want it to become DON in the gut, but would preferentially liberate DON selectively in the tumor where these enzymes are enriched. And so we made a lot. We made hundreds of these glutamine-based antagonist prodrugs. And we screened them through this paradigm. So we said, are they stable in plasma and liver? Are they stable in the gut? We were looking for something that did not generate DON there. Then we put them in human tumor cells. We looked for them to release DON. If they did, we did in vivo PK studies to look at the exposure in the tumor versus the exposure in the gut. And then if that all worked out, then we would do the efficacy experiment. So this was our screening paradigm. So let me give you an example of one of our success. One of the ones that we made was these acetylated lysine prodrugs. And this is a schematic of how that works. So here's the drug, DON, and we put acetylated lysine on it. The idea being that the acetyl group is going to come off by HDAX, which is an enzyme enriched in the tumor, not so much in the gut. And then the lysine would then come off by cathepsin L, again, enriched in the tumor, not in the gut. And the idea would be we'd release it in the tumor, and again, not in the gut. And this actually was not our idea. This had been published by another group in Nature Communications where they had used this on pure mice. And so we basically copied this idea on DON. And so I'm going to show you one of the more successful compounds from this series, a compound called JHU607. Here's the structure of that compound. But if you look in vitro, it was stable in plasma, stable in liver, stable in intestines, completely unstable in tumor. Okay, exactly what we want. And then when we put it in the presence of human plasma with human tumor cells and ask where's DON being liberated, in plasma it all stayed as prodrug, no DON, but in tumor cells we were liberating DON. So it had this profile that we were looking for. So then we took it into mice, and this is really exciting. We took it into these mice, and we, again, gave the prodrug to the mouse and looked at where the exposure of DON was. And so first we looked at tumor versus plasma, and you can see a lot of DON was distributed to the tumor, not much to plasma. The plasma to tumor ratio is about 5. But most excitingly is when we looked at the tumor levels versus the gut levels, we saw a 12-fold difference. So we were able to get 12-fold more to the tumor versus the gut, thus expanding the therapeutic index. And so this is really exciting. This is a known active drug that we've now been able to direct to the target. And so based on this, we started a new company, which is called Drayson Pharmaceutical, and we raised about $40 million starting out in order to get this into patients to make that. See if it worked like in patients, like it did in our mouse. And so, of course, the company picked a different compound and then changed the name to DRP-104 was the compound. But this compound was selected for development. And the cool thing about this compound is it is actually bioactivated in the tumor, but bioinactivated in the gut. And what happens is that here's the DON. It's got these 2 pro-moieties. Both of them come off in the tumor, liberating DON, but only one of them comes off in gut, and it's a dead end. It doesn't get metabolized anymore. So this is actually the first time I think anybody's described a prodrug that activates in one tissue and inactivates in another tissue. So this was published about 2 months ago, and it got a lot of press. So we had Hopkins picked it up, several local and news organizations, and also the NCI had a large story on it. 'Cause this is kind of cool if we can replicate this with other drugs. And so what that resulted in is that when we looked in mice, this DRP-104 compound showed similar efficacy to DON, but much less toxicity, as you'd expect. So if we give equal molar amounts of DRP-104 versus DON, both of them wipe away the tumor, okay? But if you look at the weight change, the DON animals are losing weight, where the prodrug animals are not. So when you look at overall survival, although the DON-treated animals, they don't have any tumors, but they're dying because of the GI toxicity, where our compound was complete survival. And you can see a picture of it here. Here's a normal gut showing intact mucosa and organized crypts. This is DON gut. It's pretty ulcerated. There's a complete loss of crypts, and you can see the DRP-104. It's not normal, but it looks a lot better than DON. And so to end this vignette, this is now in clinical trials. We're about to move into phase 2. So it's really exciting to have this, kind of idea in academia, and we were able to take it all the way to identify a drug and now in clinical development. All right, vignette 2. So now we're going to move on to vignette 3. So vignette 3 is about inhibiting an enzyme called GCP2 for treating IBD, or inflammatory bowel disease. And this is a drug target that we've been interested in the brain and failed, but then have shown that it's quite active in IBD. So this is a story of turning a failure into success. And this has also been in collaboration with the Crohn's and Colitis Foundation. All right, so let me tell you a little bit of the background. This is an enzyme called glutamate carboxypeptidase 2, or GCP2. In the brain, we know a lot about it. It takes this dipeptide called NAG, which is one of the most abundant peptides in your brain, and it clips this N-acetyl aspartyl glutamate. So it's a dipeptide, and this enzyme clips off glutamate, liberates glutamate in the brain, and is thought to be involved in glutamate regulation of glutamate transmission. In the gut, it's also there, much lower levels, but it exists in the gut, and it's thought to be involved in folate metabolism. Folate exists with a polyglutamate tail, and this, like Pac-Man, is taken off the glutamates one by one. It's also thought that it may be involved in regulating glutamate in the enteric nervous system, sort of your mini brain for the gut. And so my lab has been interested in this enzyme for treating brain diseases and regulating glutamate in the brain for years, like 25, 30 years. Because overactivity of this enzyme is thought to regulate glutamate and thus be involved in diseases, neurological diseases where too much glutamate or aberrant glutamate is implicated, which is a lot. So this has been looked at in numerous pain models, epilepsy, drug addiction, schizophrenia, stroke, ALS. In fact, this series of inhibitors have been, there's published literature showing in over 50 different animal models of disorders where glutamate is replicated. This compound has shown protection. So pretty exciting from a from an efficacy perspective, that this is an interesting target. And we've synthesized actually probably around 3,000 inhibitors of this enzyme. And most of our inhibitors look like glutamate because that's the active site, and they also have a zinc binding group because this is a zinc metalloprotease. And I'm just showing some examples here. This is a phosphonate-based inhibitor, so phosphate chelates the zinc. This is the glutarate part. We also have urea-based, thiol-based, hydroxamate-based. These are all potent inhibitors, picomolar potency with really nice selectivity. Problem is, as you all can predict, because they have so many carboxylates, they don't get in the brain. So the problem has been that these compounds are all polar, they're ionized at physiological pH, all the things that kind of told us we shouldn't have. We break all the rules, and because of that, they don't get in the brain very well, and because of that, nobody's ever developed this for brain disease. This has been a lot of effort, and nothing has really gone forward. So we gave up. And then in 2014, there came this paper that showed that this same enzyme we'd been interested in the brain turns out to be highly upregulated in IBD. So it's in your gut in low levels, but if you have IBD, we saw a huge increase. I'm not going to show it here, but this is about 3,000% increase. This isn't a little increase. This is a huge increase that you see. So this was the first paper. And I'm showing you here from that paper, this is brown is where GCP2 is, and you can see there's not much normally, but you see a huge upregulation in IBD. And not only is it upregulated, but they classified it as a hub gene that was connected to other IBD regulating genes. And this is the first report, but then there's been 6 other labs that have shown the same thing, so this has been replicated. And so we thought, all right, we have these drugs, they're super potent and selective. We can't get them to the brain, right? But we could get them to the gut. That's a lot easier to target. And so we basically took one of the tool compounds, 2-PMPA, and we put them in a variety of IBD mouse models. And they worked. And I don't have time to go through all the data, but there's now multiple publications by our group and others showing these compounds, at least if you're a mouse with IBD, we see positive effects. But the compounds that we used were these public domain tool compounds. This compound called 2-PMPA, you can order it from Sigma or Tokers. And also it was active when we gave it as an enema locally. It has very poor PK. And so we took this to the Crohn's and Colitis Foundation, and they funded us to develop an orally active, gut-restricted GCP2 inhibitor. And so I'm going to talk a little bit about this. Again, we made probably 100 of these prodrugs and conjugates and tested them. And I'm just going to tell you about the lead. So our clinical lead is a compound that is deoxycholic acid linked to 2-PMPA, and I'm showing the structure here. This conjugate is a potent GCP2 inhibitor, so 4 nanomolar. It's very well tolerated. We've now done non-GLP dose ranging in up to 1,000 milligrams per kilogram, 14 days. We don't see toxicity. It's also gut restricted. So when we're giving it orally, we see 50-fold more of the drug in the gut than we do peripherally. So we have a gut restriction. I'm showing here now. This is the jejunum and colon, and then plasma. It's hardly measurable. So with this compound, we have now tested it in multiple animal models of IBD. This one I'm showing is a DSS murine colitis model, and we tested it against 2 clinically used drugs, one being sulfasalazine, one being tofacitinib. And in both cases, our drug had a better effect than the 2 compounds that are in the clinic. This is DAI, this is the disease activity index over time. And so you give these animals DSS, which inflames their colon, then you give them the drugs, and you ask if their body weight, bloody stools, diarrhea, there's a bunch of things that make up that daily index, but if it improves with your drug. And what you can see is when we treat with our drug, we see a statistically significant improvement in both cases, better than sulfasalazine and tofacitinib. And when we're giving this drug, we see complete decrease of that elevation of this enzyme. Right, so it's really highly upregulated. We see a complete inhibition of that, so we're seeing target engagement. And the last is, remember I told you we studied this in the brain for a long time, and one of the areas was pain. Right, we were seeing this analgesic effect in pain, and so one of the questions we asked is, this drug that we can now get to the GI, does it have any effect on GI pain, on visceral pain? Because that's a very unmet medical need. And so in working with Jay Pastrika, who's a professor in GI at Hopkins who studies pain, he did a model of analgesia in post-inflammatory chronic visceral pain and showed that we had complete normalization of the pain response in this animal model. This is a TNBS animal you give to the animal, and then you implant electrodes and you do a colorectal distension, so you just stent the colon, and these animals are hypersensitive to that, and you look at their visceral motor response. And so animals that have had TNBS versus vehicle here are hypersensitive, have an elevated visceral motor response. But if we give in this case, 10 or 1 milligram per kilogram of our drug, we're now going lower because they all completely eliminated. We see a similar response than 300 milligrams per kilogram of gabapentin, which was the control. So this is pretty exciting. And so right now we have offers from both big pharma and from a venture group that want to start a company. And so I'm working with Johns Hopkins Tech Ventures right now to see which way we should take this program. But hopefully that gives you an example of a project that we worked on for years and failed and couldn't get it, but ended up turning that to a different therapeutic area with what at least at this point looks like to be successful. And I'll end by just thanking the people that did the work. So this is my lab. This is Johns Hopkins Drug Discovery. Ajit and Takashi Tsukamoto here were very involved in the System XC project. Let's see, Rana Race, who runs our DMPK group, and Katie Lemberg, who's a physician scientist in oncology, was very involved in our glutamine antagonist project. And then Rana and Diane Peters, who runs the IBD project in the lab. So anyway, I'll stop there. And first, I'll appreciate the funding sources that did all of this. And then I'll stop there and take any questions. Thank you.
Questions for Barb?
Actually, nice talk. Actually, XCT, it's one of my targets for drug addiction. But so, for
all right.
Coming back to the vignette one, so I'm worried about inhibitor. You are inhibiting the XCT, but the question is, that's going to go directly to the tumor or maybe other brain regions? And you don't want that, correct?
So the XCT project that we've licensed to AZI was not directed in any way. The compound had very good brain penetration, but it was not selective to the tumor.
Right. So it can now for for vignette 3, where you talk about
GCB2 and IBD.
Correct. Is that something that can be used for drug addiction to modulate glutamate?
So there's probably a half a dozen papers showing that inhibiting this enzyme is good for drug addiction. Again, the problem is that these work in these animal models by giving these huge doses because only about 1% gets to the brain. And so nobody's going to take that forward clinically, right? We got to get a better drug. So if you're a mouse and we load you up with a lot of drug, it'll work. It absolutely will work. Or if we inject the drug into the brain, it'll work. But the drugs that we've developed are not are really just not going to be able to move to the clinic because of their poor pharmacokinetic property. They work. Okay. Again, mouse, lots of dose, it works. But clinically, I don't think it's going to go forward.
What's the log P? Do you know?
I don't know. Okay. Bad.
Well, I will send you an email.
Okay. And if you want some inhibitors, we can talk afterwards. Yeah, absolutely.
Hey, Barbara. This is a
Yes.
fantastic talk.
Thank you.
I have a general question. When I was a postdoc, we collaborate with a company. I heard one major complaint is the company, they don't want to publish, right? Like that's a constant battle between the research lab who want to publish, the company, they don't want to publish. Is that situation being improved? Like, did you just, when you formed agreement, you want to emphasize that we want to publish? Like how you handle this situation? Thank
So
you.
at least in my institution, we cannot have any agreement with a company that does not permit us to publish. So that's just ground rules. Typically we, if we want to publish, we have to give it to the company and ensure that there's no patentable material, and if there is, give them time to patent. But honestly, we do that anyway. We don't publish things without first patenting, so it has not been an issue. I think one of the issues, I didn't talk about this, but it's in the high throughput screening agreement that we did with ASI. One of the issues that Hopkins had a real problem is here we are screening this company's library, right? And then giving it back to the company to develop. What's academic about? How are we increasing knowledge? And so what we ended up doing, which has turned out to be really cool, is we did the screen and we purposely select a lead series that is gonna be public domain. We're going to do SAR, we're going to get it to the target, but it's going to be scaled up and shared with anybody that's interested. And then their other group of compounds, they are going to be more proprietary and be developed by the company. So that was very purposeful to make sure that we were doing our academic mission. Does that make sense? Yeah. So that agreement took about a year to to finalize, but that was all regulated up front. In all cases, we are allowed to publish, given the company's ability to review it first. Yep.
Hi.
Hey, Kelly.
So you're kind of at the decision point right now in Vignette 3 with which way to go. And so in the first one, it sounds like you knew you wanted to go the industry route and tried a bunch of companies and then went with the one that said yes. And then in the second one, it sounds like you formed a company. And so I'm wondering at each of those points, like what guides the decision-making process and like, so you're actively doing it right now and how, like what guides that decision-making process?
Yeah, that's a great question. So in the first vignette, I really wasn't looking for a partner to take the drug forward. I was looking for someone who would give me a decent library. And so that was pharma. Right. I'm not going to get that from a VC or a small company. That was pharma. And so that's what we were looking for, and so we approached big pharma. The second vignette on the glutamine antagonist is kind of risky. The compound's a diazo. It irreversibly inhibits these enzymes. That's a little risky for most of the big pharma companies. And so we decided to go a small company route to get to the clinic to show some efficacy. Ultimately, it'll go to pharma for development if successful. In this third one, I think either route would be good. We're really looking at what our best deal is at this point.
Nice.
Yeah.
Yeah, thanks for a really interesting talk. I was wondering, so kind of similar to Kelly's question, you know, when you're looking at potentially partnering with an industry and kind of going through that whole negotiation process, how much of that do you have input on as the, you know, principal investigator at your institution versus is this just done with like lawyers through your tech transfer group?
Yeah, so Tech Ventures for sure is leading it. I probably have a little bit more input than normal because of my background. Actually, when I was in pharma, I was responsible for academic collaborations. So I've been sitting on the other end of the table, and so I think I've had probably more say than most. But this is definitely decisions are by Johns Hopkins, not me. I mean, I can be advisory, but it's their decision of how to license it out. It may be different in different universities. I don't know, but that's my situation. Yeah.
Thank you. It's a very fascinating multiple project collaborations. It's very encouraging. So I guess my question is that when you approach the industry, how to walk the fine line without exposing too much details?
Say again?
How do you walk on the fine line without talking too much details so they know everything when you approach the industry so that they really trying to talk to you and start collaborating rather than they taking everything work on their own?
Yeah, so I mean, typically when you first have a conversation, like a dating, where you don't give all your secrets away the first date, right? So you typically we put together a pitch deck that's pretty simple and talks about the idea. We do not approach anyone, VC, pharma, unless we have IP protection. Okay, so that's first. And then when you go out, you make a small pitch deck, maybe 10 slides. I don't want to see any more. They see a lot, so that they see if they're interested. Then if they're interested, and then you'll have like an hour meeting, and we go through more of our data. And then if there's interest, then we have 3-hour meetings and all that. So it's a process. But number one, you don't want to have any conversations until your IP is protected. And then number two, you start off, giving a little taste or bite of the project and see who's interested. And then, more detail after that.
Thank you. So I have a second question, which is more about science. So when you trying to inhibit the system XC
Yep.
on your cancer model, do you also look at the ferroptosis? Because that's
Yes, in fact, we've had a couple of papers with Brent Stockwell's lab looking at these and ferroptosis. I can say the A's ideal is about 5 years old. I pulled it out just because I think it's an interesting example of working with pharma, but they have taken it. At this point, we're not doing that. We're not really doing that project anymore.
Thank you. Thank you for going through some real life case vignettes, because I think that's really when you unfortunately learn what not to do. So I actually have a question, and it's a real life going on right now. We've engaged with our academic institution and a small biotech and started working, but it's written up as a contract to say, they're going to provide support. And what happens if and when that relationship, they decide, we're not going to continue studying this. We don't want to shelve it, right? But how do we kind of go back? And not that I'm trying to be negative about relationships, but if it doesn't continue with that, are you able to go back and continue working on your strategy?
Yeah, first of all, most things fail. Okay,
Exactly.
so I'm showing you the 3 examples that worked, but I can show you 10 more that came back to us. First of all, when we negotiate, when Hopkins does the negotiation with pharma, we have, I don't know, clawback rights, I'm not sure if that's what it's called, but if the company doesn't continue working on it, all the rights come back to Hopkins and we can try again or take it on ourselves. So that's how it's done. So we would never sign a deal that says they take it away, and if they stop working on it, actually, NIH won't let you do that either if they've funded the research. So that's always in the contract, that if they don't use normal efforts to develop it, it would come back to the university. Yep.
How do you determine, and at what point do you determine what is intellectual property worth protecting? I mean, sometimes projects develop, in academic labs over many years, and you publish and put in the public domain bits and pieces of things that kind of ultimately inspire the idea for a drug discovery program.
Yeah,
so how do you actually determine when and what to
Yeah, so
protect?
unfortunately, ideas are tough to patent. I think the most valuable patents are the composition of matter patents. This is the product patent. So those are the most valuable. Sometimes we will do a composition of matter patent, and on the same day we do a broader use patent, reducing it to practice with several compounds. So trying to get a broad coverage. At the end of the day, the composition of matter patent is gonna hold water. It's gonna be most important. So for us, the patenting is, I'd say probably 75% is composition patents.
This raises a really interesting question, because it's the chemists who have most of the intellectual property. And since often there's a financial benefit to having that intellectual property, it can be a little bit unfair. So I think some institutions have begun to discuss the possibility of incorporating a larger group of people in the benefits that accrue to having intellectual property.
We talked about this at our table. I think, is Jim still here? And Greg? Yeah. I think the Lieber Institute has an IP policy where they have inventors that are defined by USPTO law of inventors, but then they have contributors. And so if any money happens to come in, it goes to both the inventors and the contributors to make sure what you're saying, Mike, they tried to mitigate that potential. I don't know, Greg, if I got that right.
That's exactly it. Yeah, there's provision for non-chemists, because as a non-chemist, as we said, it can seem unfair sometimes, but there's a key contributor provision for people who aren't chemists to.
Well, I want to clarify, because it's not just chemists that are on these patents for sure. I'd say half of the ideas come from the DMPK group that tell the chemists what they need to do because their compounds are being metabolized. And then who reduces it to practice? So if you get these compounds and you show that they work, show that they have decent PK, alter the the metabolic process, in our group it's typically in the entire drug discovery team that's been inventive in the process. But
So very quick. Sorry. So if you have a compound, let's say you have a lead compound, you went through it. What do you need, and how you can find the company? How much it's enough? You have lead compound. How much it's enough to convince the company, and how you find the company? That's where I am now.
I'll start with the first question of what's enough, and that changes with PCs and pharma all the time. It's like, probably 6 years ago they were taking ideas forward into companies. I think today you need a little bit more beef behind that. So that does change. I think your tech venture office, your Office of Technology Transfer, it's part of their job is to go out and find people. I'd say in my case, just is a little bit different just because I have a experience in pharma, so I have a lot of contacts already that we pitch to. So most of the companies that we've worked with have come from that, not from the Tech Ventures. Although Tech Ventures is very active, and I mean that's part of the job is to take the IP and find and find takers.
And maybe what I could offer is that most pharma companies have search and evaluation groups, and their job is to go out and look at academic institutions, small biotechs, or receive pitches that you might like to propose. And they will assess them against what we're interested in from a particular therapeutic area point of view. If we like what we see, then, as Barb said, maybe first all we've seen is non-confidential information. We might sign a CDA which says whatever you show us, we're not going to disclose to anybody else, and then you show us a little bit more. And then we might actually do a deep dive do a whole diligence, review all of the experiments, all of your reports, and then decide whether we would like to join in a venture with you.
Actually, the one thing that I didn't mention is that a lot of these external innovators from pharma show up at meetings, and so if you're presenting at meetings, they're there, and if it's of interest, they may approach you there. That's another way we've. So scientific meetings. So whatever scientific meetings you go to. There's pharma. Is it still called external innovation? So those that are basically looking for innovative projects that would fit the company, they go to these meetings and listen to presentations, and that is another way to get partnered.
No, I would say what I published was out.
Yeah, that makes sense. Yeah. Okay.
Okay, I think we're
All right.
good.
Thank you.
All right.